Hey AP, hope you're doing well when you watch this video. So the goal of this video is to help you review organelles. Again, organelles are covered extensively in biology, but just in case you need a fresh reminder, I hope this helps. So let's go ahead and get started. Let's begin with the nucleus and ribosomes. So ribosomes translate the nucleotide sequence of a messenger RNA molecule into a polypeptide. That's a protein. And so remember, the central dogma is DNA is converted into RNA by the process of transcription, and then that's made into our protein, which is translation. Okay, so ribosomes are protein manufacturing factories. They occur both in prokaryotic and eukaryotic cells, and they consist of different things. They consist of protein molecules and ribosomal RNA. So they consist of both. So this is a scanning electron microscope, well, actually a transmission electron microscope of ribosomes. So these, you can see the ribosomes are in the middle right here, and they're making proteins. You can see that. So they're not membrane bound. Organelles and eukaryotes, they're free in the cytoplasm, but they're also attached to the endoplasmic reticulum. And that is called the rough endoplasmic reticulum because it looks like it's studded with beads underneath a microscope. Of course, in prokaryotes, they float freely. So the nucleus is usually the largest organelle. We saw that when we did our cheek cell slide. You know, that dark circle, that was the, we had our cell, and then we had like the dark circle in the middle, and then maybe a few lighter circles. So that is the largest organelle. It's a location of DNA and DNA replication. Again, your cells have to be copied, so DNA has to be replicated. It's a site where DNA is transcribed into RNA, and it contains the nucleolus. The nucleolus is a region where assembly of ribosomes from RNA and protein begins. Where assembly. So we actually start building those ribosomes. So the nucleus is surrounded by two membranes that form a nuclear envelope. So you can see here, here's the nucleolus region, chromatin, nuclear envelope. There's openings, of course, because we have to have molecules between the nucleus and the cytoplasm, like DNA, RNA. When it's transferred into RNA, that RNA has to exit. So in the nucleus, DNA combines with proteins to form chromatin, and those obviously form our chromosomes. The outer membrane is continuous with the ER. So the endomembrane system, I would definitely know this. I would definitely review it because this is a very important system. It includes the nuclear envelope, endoplasmic reticulum, Golgi, and lysosomes. So tiny membrane-bound surrounded vesicles shuttle substances between the various components. So these are kind of, like suitcases in a way. They shuttle various things. So here is a picture here, and I would be able to walk yourself through this picture, okay? So here's the rough ER. We're making proteins. Those proteins are released in a vesicle, okay? Membrane-bound vesicle. So they are protected. And they fuse with the cis region of the Golgi, okay? The Golgi is like the post op. It modifies, adjusts, and then ships them to the correct place. So the Golgi targets them to the correct destination. Sometimes the proteins are meant to be in the cell. Sometimes the proteins for use outside the cell. There's various different reasons. The smooth ER, the rough ER and smooth ER are different. Rough ER makes proteins. The smooth ER, though, is a site for lipid and steroid synthesis, and glycogen breakdown, and chemical modifications of toxins and calcium ion storage. So be able to contrast those two, the rough ER and the smooth ER. So again, the endomembrane system, it's important because it regulates protein traffic and performs metabolic functions. Know what it contains. And here's just another picture of it. Now moving on to the smooth ER. So again, this is a transmission electron microscope picture. You can see the smooth ER is here. Doesn't look studdy. You can kind of see the studs over here, hopefully, with a rough ER. And so whatever its functions, it modifies small molecules such as drugs. So it modifies. It breaks down glycogen. It also makes lipids, which is a very important situation, and steroids, okay? So I want to ask this question. I want you to pause the video if you so desire and try to answer it before I go through the answer. Barbiturates, alcohol, and many other drugs induce the proliferation, which means the rapid growth of smooth ER and its associated detoxification enzymes, proteins, thus increasing the rate of detoxification. So what does this mean for the user then? Pause. So the answer is, this is the beginning of addiction. You start let's say you drink a couple of drinks and you feel the effects of the alcohol, but over time those drinks don't have the same effect. You've become used to it. You've adjusted to it, because the cell doesn't know what you're doing. The cell is just like, hey, this is poison. Let's protect the body. And so it grows more and more, and it proliferates and makes more enzymes to help you break down this toxin faster. But that leads to dependence and you have to increase your levels of the drug to get the same effect. So the rough ER, you can see right here, makes proteins. An example would be insulin. These proteins are transported in vesicles. Again, endomembrane system, know where that is. So they're transported, they're made. Many are linked to carbohydrate groups. We'll talk about that when we talk about the cell membrane. Glyco, sugar, proteins. And those are important. These proteins that are linked, they're important because they help identification. Your cells know you versus not you. And how do they know that? Because of carbohydrates on the outside of the cell. Here's a picture of the Golgi. Again, it kind of looks like a stack of pancakes in a way. And the Golgi is sort of like the post office. It receives and dispatches transport vesicles. So it modifies proteins inside, maybe adding a methyl group here, hydroxyl group there, you never know. And then it transports them to other cells and it ships them, it targets them for its destination. And so it packages, sorts, and sorts proteins, adds carbohydrates to proteins, and then it ships them to the right address. Lysosomes are kind of like the garbage places, if you remember, stuff like that. So lysosomes, they have digestive enzymes right there. And so hydrolytic, which means it breaks down using water. And so lysosome fuses with a food vacuole, and then those vacuoles break down that, so digestion happens in lysosomes. Sometimes lysosomes also break down damaged organelles. So the cell really cleans up, when stuff is damaged. And this is just an example of a way that happens. So you have phagocytosis. Phago means like eating, so the cell's eating, bringing stuff in. And so you have your lysosome, it fuses with it, and it releases the waste. So macromolecules may enter the cell by phagocytosis, and then they fuse together with lysosomes to break it down. And this is just another picture of that. Lysosomes is generated by the Golgi. So the Golgi has all those proteins inside of it, and then you can see how it fuses together, and digestion happens, and then we release those byproducts for excretion. So let's ask ourselves a question. Explain how the pH of the lysosome, in which enzymes have an optimal pH in the acidic range, could decrease. So pause. Now I want you to start thinking about hydrogen ions. And so in order for the lysosome, this is the lysosome, in order for the lysosome to decrease in pH, you have to pump hydrogen ions into it. So that would involve some sort of pump, protein pump, and there's theoretically other areas that could do that, but you have to be able to pump it across to lower the pH, because the more hydrogen ions, the more acidic a solution is. So in eukaryotes, breakdown of energy-rich molecules begins in the cytosol. The partially digested molecules, again, energy-rich, let's just think glucose for a second, enter the mitochondria. And everyone remembers the mitochondria. If I could draw really good, I'd draw a bicep right now. But it's a powerhouse of the cell, right? We're converting chemical energy into ATP energy. Again, energy can't be created nor destroyed, so we're just converting it from one form into another. Cells that require a lot of energy, thinking like liver here, heart cells, they have a lot of mitochondria. So mitochondria have two membranes, outer and inner, very porous. Of course, we gotta get stuff in and out. And then there's extensive folds. Okay, so here's a picture of it, outer and inner membrane. You can see there's ribosomes in there, the matrix. This is a transmission electron microscope, 1 micrometer, so we're crazy zoomed in there. So plants and algae cells contain plastids. Another organelle they're called plastids. And that can differentiate organelles. Some are used for storage. One example is the chloroplast. contains chlorophyll, the site of good old photosynthesis. And so again, we're converting energy from light into chemical. So it's an anabolic process, which it's an endergonic process as well, right? We're building anabolic steroids, we're building. And so chloroplasts have two membranes plus internal membranes called thylakoids. And so there's granum, which is a stack of thylakoids. So I think like green pancakes, right? Thylakoids, granum is a stack of green pancakes. And then the stroma is the aqueous matrix around the granum. So if you have your pancakes stacked, that's a granum. And then you have the liquid around it, that's your stroma. Okay, and so you can see a picture here. Thylakoid, there's your stroma right here. The granum is a stack of them. And we'll dive into exactly what happens in photosynthesis later on in this course. Here's an example of plastids. Sometimes plastids can store, like colors. Where do colors are stored in plastids? So you can see there. Sometimes we store starch in plastids. Again, I wouldn't know these special like leucoplasts or chromoplasts. That's excessive, but just knowing that plastids store this stuff. Peroxisomes is an example of oxidation. So it's a good example of how compartmental structures are crucial to its function. So it removes hydrogen and adds oxygen, forming hydrogen peroxide. Then there is an enzyme, good old catalase, we already dealt with that this year in our lab, that breaks down hydrogen peroxide into water. Vacuoles store things, materials, but sometimes they can also functionally maintain homeostasis. So freshwater protists give rid of excess water by contracting. So they bring the water into the vacuole and they contract it and excrete it from the cell. So it enlarges as water enters and then quickly contracts. We'll skip forward. And then finally, the cytoskeleton. Cyto means cell. Skeleton supports you. Again, we're just a pile of flesh and meat on the floor without our skeleton. And so I would definitely understand the cytoskeleton, very important. Supports and maintains a cell's shape, holds organelles in position, moves organelles. You remember the walking protein in the video I showed you? The walking protein. It can transport cells in the protein. Involved in cytoplasmic streaming and interacts with extracellular structures to anchor cell in place. So a cytoskeleton is made of many different structures, such as microfilaments. Microfilaments help a cell or parts of a cell to move. So they determine the cell shape, and they're made from actin. Okay, this is a protein. So microfilaments are made from actin. Intermediate filaments anchor cell structures in place, resist tension, maintain rigidity. And then, so here's an example of that, intermediate filaments. Maintain cell structure. Again, they label it with a radioactive dye. And then microtubules made from tubulin, they act as a framework for motor proteins to move structures in a cell. Those are your walk-in proteins, which is insane how God made this stuff go so complicated. But walk-in proteins to move structures in a cell. So there's your microtubules. So this kind of sums it up. And if you were to review something, I'll show you what to review right here on this slide. Actin filaments, intermediate, and microtubules. What I would know is that actin and keratin and tubulin, all those are proteins. Okay? So they're made of different proteins. But what's important here, actin can move cells and maintain cell shape. And it also can divide animal cells in two, which is important. This intermediate filaments is all about maintaining and anchoring, okay? And then microtubules can move chromosomes, I would know that, and also organelles by those motor proteins. Those are the things that I would know on this slide as far as cytoskeletons goes. Microtubules also allow for cells to move cilia and flagella. Cilia, we have cilia all over our bodies. Flagella, there's only one cell in the human body that has flagella, and that's the human sperm cell. And so cilia right here, you can see short, usually many present. So if you were to try a cigarette for the first time, you would cough because the cilia in your respiratory system would try to move those toxins out of your body. I don't know if you ever had a violent coughing spell, you can breathe in dust or anything like that. So here's, cilia. They have like the radial spoke and then the proteins around it. They kind of move in like an oar-like motion. There's many videos you can watch. And so there's many different ways to understand how cell structures are created. So Plant cell wall is the next one. I skipped a few things just because for time reasons, but plant cell wall, rigid structure outside the cell membrane. So again, definitely protection. It can link with other plant cells near it. So you can see right here, there's a cell membrane on the inside, there's a cell wall, and then there's the lamella, and then cell wall, and then membrane, okay? So it has three major roles: support, barrier to infection, and contributes to form during growth and development. And so there's times when plant cells are connected, and what are they connected by? They're connected by channels called Plasmodesmata. These channels allow movement of water ions, small molecules, hormones, and some RNA and proteins. So many animal cells are surrounded by a matrix. Just like plant cells have a lamella, animal cells have a matrix that surrounds them. And so you can see right here in this picture. You can see the cells, kidney cell, blood vessels. But right here is the extracellular matrix. And so what's in it? Well, there's proteins, polysaccharide chains that provide strength, that keep stuff in place, helps filtering as well. And so it holds cells together, helps filter material as well. And then finally, the last thing I believe for this is cell junctions. How cells are attached to other cells. There's tight ones, desmosomes, and gap junctions. So tight junctions prevent substance. There's nothing. Desmosomes hold cells together but allow materials to move in the matrix. And then gap junctions are channels that run between membrane pores and adjacent cells, allowing substances to pass between the cells. So here's an example here. Tight junctions, nothing's getting through. Desmosomes hold cells together but allow materials to move, few materials. And then gap junctions are kind of like small little channels. You can see right here, tight junctions, and desmosomes. They do permit materials. Again, they link adjacent cells. And then gap junctions let cells communicate. How do they communicate? Through cell signaling, which we'll talk about later on in this course. Cell signaling. Again, an example of why that is important, let me just show you one right here. I've had some heart issues in my life, but rhythmogenic right ventricular dysplasia, ARVD, is a very dangerous inherited myocardial disorder associated with arrhythmias. And so literally this arrhythmia is because a protein, desmosomal protein, isn't made correctly. It's mutated. And so because of that, the cell signaling with hearts can be interrupted at really dangerous times, especially in high activity moments. And so this ARVD is responsible for many sudden cardiac arrests during athletic events, sudden death. And why? Because a protein is mutated, and therefore cells cannot send the correct signals correctly, communicate correctly, and therefore leads to arrhythmia. And I'm not sure what just happened. Okay, so I hope this was helpful. I hope you're doing well. I'm so proud of you for the hard work. I know it's a lot. I know all the AP classes together add up to a lot, but I am so proud of you. Don't give up. God bless, guys.